Hi, welcome to Chapter 12, Skeletal Muscle Function. We will focus most of our study time on voluntary skeletal muscle function, and then we'll move on to involuntary muscle function, including our mini cardiac unit within the topics of muscle, since we have 3 muscle types we need to learn. But most of our time will be focused on skeletal muscle function. We want to think back to the action potentials and the nature of an electrical impulse that we worked on so hard in Unit 2, and take this idea from Unit 2 of an electrical impulse used to stimulate all 3 muscle types as we go through them. They all need to have that initial depolarization to start contraction. And those are some of the things I want to go through with you guys in this lesson. So our modified learning goals are going to be to identify anatomical muscle structures, identify muscle cell structures and associated function during contraction, and then describe the sequence of events during muscle contraction, including relaxation, where the cycle could start over again. Muscle fibers look like tiny bundles within a bundle. You can see the somatic motor neuron synapsing with the surface of the fiber at what's called the neuromuscular junction. So this is a special type of synapse. This junction is where the action potential ends and the chemical ligand acetylcholine gets released. If we peel back the sarcolemma, a fancy name for the plasma membrane when you're talking about muscle, we can then visualize the cellular machinery inside this fiber. Skeletal muscle fibers have an extensive network of sarcoplasmic reticula, or SR. This is needed for calcium storage and calcium release. This action happens during a contraction. The picture I want to paint for you today is the theory of excitation-contraction coupling. This was awarded the Nobel Prize in 1963. There are a few key structures that allow this type of function to happen. If we zoom back out using our microscope and we look at a microscopic view of skeletal muscle, we see obvious vertical stripes called striations. This striated pattern is characteristic for all skeletal muscle fibers. This pattern is due to the alternating proteins called actin and myosin. If we go back now to figure 12.2, we can see the neuromuscular junction a little easier. This is where the action potential causes the release of acetylcholine. 100% of the time, this chemical ligand is going to be excitatory and will depolarize the sarcolemma to threshold. To coordinate the depolarization of the entire muscle, the T-tubule, shown in green, extends down from the surface of the sarcolemma into the bulk of the fiber. T-tubules act like an elevator shaft during the depolarization event into the entire muscle fiber. Notice the connection between the SR, shown in purple, and the T-tubule itself. They touch each other. They're in contact with each other. They're in contact with each other through DHP and voltage-operated calcium channel proteins located along the length of this interaction between the green and the purple. Now let's talk about sarcomeres. Sarcomeres are the smallest functional units of muscle and the smallest measurable unit for contraction. It's part of the myofibril between 2 Z-lines. A Z-line is formed every time the actin proteins anchor to each other, and it looks like a zigzag line drawn here in blue. The thick myosin filaments float in between the blue actin lines, and they're shown in red. These are all the horizontal lines going back and forth in this single myofibril shown here in figure 12.3. So the sliding filament model for muscle contraction starts with excitation of the muscle fiber through motor neurons and using the sarcoplasmic reticulum and T tubules together to release a large amount of calcium into the ICF compartment, which triggers the actin and myosin protein filaments to interact with each other through cross-bridge formation. When calcium is released, it must find and interact with two regulatory proteins that can be found along the blue actin protein strand. These regulatory proteins are troponin and tropomyosin. Cross-bridge formation, shown here in step number 6, down near the bottom, in your figure 12.8 from your textbook, is where the force is generated. Okay, so the proteins can interact with each other through cross-bridge formation. This force is going to cause the sarcomere to decrease in length and shorten overall. Please refer to the Pearson website with your student login to view the AP Flix on this topic. That's a light blue link. It's very tiny. You'll have to click through it twice once you find it in your toolbar on the left-hand side of the Pearson website. You should expect a lecture quiz based on the Pearson videos as well as these found here on Canvas.